All right, here we go. All right, everybody, welcome to Bio 30. This is week 2, and I was just telling the students that were here about these notes. I need if you don't, this is new for me, new to you. I haven't used them before. Hopefully, this will help you follow along and make it helpful, and then give you something to study off of after, something to make your flashcards with. And just in general become a better student as you go forward in learning. When you go to university next year or years after, a lot of note taking, and this is another skill that you just need to start honing, and again preparing yourself as you go forward. So here we go. So print those off, pause it right now if you're watching it, print them off, and then go. All right, let's go. All right, so where should I be? So at this moment in this course, where should you be? Well, you should at least have started by now. So lesson 1, lesson 2, those should be completed. You should be looking to be done relatively soon. The first due date is module 1 is due on the 30th. It's the 22nd right now. So that basically gives you 8 days to finish. So if you haven't started, time to get going. Let's move forward. 8 days, that is basically a lesson per day. If you do that, I think there's 8 lessons, so that's a lesson per day if you haven't started. So now, since maybe you've procrastinated, maybe you had other things going on, I don't know, but due date's coming up on the 30th. For those who haven't started, I'm going to be contacting you if you are, and your parents if you're under 18, and just to make sure everything's okay, because at this point everybody except for a couple people have been in the class for 2 weeks, so there's no reason that you shouldn't have started by now. When I'm in a course, when I'm doing my lessons, what should I focus on? Well, I should be focusing on the content, so I would read the content. If it tells you to read something in the textbook, I would read it, and then watching the videos. And as I'm going, I am making notes, I'm making flashcards, because these flashcards are super important as I'm preparing for my exams and eventually the diploma. The diploma being worth 30% of your mark, and the midterm and final both being worth 20% of your overall course grade. Now I've mentioned that now we have these notes that I have to prepare. I'm doing it week by week. So next week 3s, I'm going to be working on week 3s this week after I'm done this lesson. So I won't have anything. I probably won't have anything advanced unless I have a lot of time on my hands. Typically, I do not have a lot of time on my hands, so I'll be making these notes for you for next week. All right. So let's move forward, and here we go. We're doing the nervous system. So the second part of the nervous system, we started the nervous system last week, and we looked at the brain and how it is, and how the nervous system is broken into different sections and how it all works. So now we're going to go into a little bit finer details, and we're going to dive right into the neuron and how things are fired. All right. Whoa! Living cells can only survive and function within a very narrow range of conditions. Okay. Homeostasis, something that you're going to be talking about throughout the course, is the body's ability to keep normal body functions. Okay, the internal environment in the same stable state in order despite variation in external environment. So basically, it gets hot, your body regulates to make it cool down. It has things built into the body. If the body's cold, it has regulations to make it warm up. So the shiver reflex is an example of, if you're cold, what the body does, it vibrates quickly in order to generate heat. So that is an example of homeostasis. Okay. Human survival depends on a system of communication between the internal environment, so in your body, and the external environment, so the things that are around you. Okay, both the nervous system and the endocrine system hormones, okay, perform this function. So the nervous system, so your brain and everything inside that tells your communication, that does all the communication, and the endocrine system. So things like the hormones do all this. All right, its responses to internal and external environments are made possible by the electrochemical messages relayed from the brain and the nervous system, or by the chemical messengers, hormones or blood. So chemical messengers, endocrine, nervous system, the brain. All right. The main functions of the nervous system. Okay, so what are the main functions? The main function is to receive information and carry it to the central nervous system. So it's relaying information. And interpret impulses and make decisions about the response, and sorts impulses and sets priorities for action. Okay, so those are three things that it does. So receive information carried to the central nervous system, interpret impulses and make decisions about the responses, sorts impulses and sets priorities for action. So like, for example, walking across the street or eat a bug or get hit by a car. Okay, and then it transmits impulses to the motor units, muscles, which carry out appropriate action. So the items that for your notes, just to clarify, things that are underlined are things that you need to write down. Okay, I know I'm going quite fast. You may need to go back and pause it and watch the video just so you can slow it down and in order to write everything down. So if you do miss it, make sure you go back. All right. All right, so nerves. There are two main types of nerve cells found in the nervous system. One is glial cells, all right? Neurological cells, which are non-conducting and used for structural and nutritional support. All right, so that's what a glial cell looks like. It's got, you see its dendrites and its axons and all these awesome things with the nucleus in the middle. All right. And then the neurons, so the neurological cells that conduct or carry nerve impulses. All right. And so here's what a neuron looks like. And we have the dendrites that are identified. We have the cell body. All right. The nucleus in the middle. And we have this long word here. This is the axon. And then the synapse, where the information is transferred from one neuron to the next neuron. All right. Neurons are the basic unit of the nervous system. A nerve is actually a bundle of individual neurons. So basic unit, nerve is a bundle of neurons. So there we are. We got a bundle there, right? So that's a neuron, a nerve. Neurons are composed of a cell body and a number of branch-like extensions. All right. So here is another picture of what a neuron looks like. Okay. We have, again, the axon, right? So that comes down here. These are the myelin sheaths. We'll get into those later, or a little bit later. They make things go faster, or transmit the information faster. We have the dendrites over here on the side, nucleus, and then we have the axon terminals. So the terminals are at the end, and then it transmits the information onto other neurons. Okay. Again, the cell body, this is also known as a soma, right? So you'll hear that as well. So the presynaptic neuron. So a presynaptic neuron is as that information is coming, and then post is after. So information comes pre, post. All right. Okay, components of a neuron. So we have the dendrites, or short thread-like branches that surround the body of the neuron. Okay, I pointed those out on the ends, right? Dendrites carry received nerve impulses towards the cell body. So it brings it towards the cell body. Axons are longer branches that carry impulses away from the cell body. Okay, so dendrites bring it in. So that's why we saw them on the end. So the dendrites bring it in. Axon brings it away. All right. Axons may be enclosed in a fatty sheath called the myelin sheath, which I mentioned. Okay, so the myelin makes up the Schwann cells with small gaps, each Schwann cell called the nodes of Ranvier. Okay, so myelin makes up Schwann cells. So those are Schwann cells. Myelin makes the Schwann cells. So are myelin and Schwann cells the same thing? Well, no, myelin makes Schwann. It's like saying because you use dirt and water, is that concrete? No, but that mixed together, that makes concrete, if that makes sense. All right, because of this, the myelin sheath increases the speed of the nerve transmission. Okay. The myelin acts as insulation for the neurons by preventing the loss of charged ions. Okay, so the myelin acts as an insulator and inhibits ion movement from the axonal membrane that it surrounds. So the information comes here, boom, it gets pushed, it's insulated, it makes it move faster. All right, so here's another picture. Why did I do that? All right, so as you can see in here, we have the myelin, okay, the myelin that's making up the Schwann cell, okay, and then we have the Schwann cell that is around the axon, and then in here you can see these right here. So those are the Schwann cell nucleus. Okay, so axon in the middle, myelin surrounding it, which in turn makes the Schwann cell. Okay. Wow, that's annoying. I get rid of that. Okay, so again, here's another picture of the neuron. So you can see how many times I've shown you the neuron. It's important that you know it and you know all the little information that goes around with it. Okay, so again, we got the cell bodies here. We have the dendrites, which are the receivers. It takes the information, sends it to the cell body. Then the cell body sends it along the axon. If there is myelin, it conducts it faster. Think of it like it's hopping. In between here, we have the node of Ranvier. So that's a node of Ranvier. So it's not just that one, it's that one, it's that one. So the space in between. Okay. And then the insulating fatty layer that speeds up the transmission. So think of it like information comes, boom, jumps, boom, jumps, boom, jumps, boom, jumps. Okay. And then we have the Schwann cells, right? And those are the nucleus right there. And the information gets down the axon terminals, and boom, it gets sent to the next cell body, which takes it in the dendrites, and everything repeats itself. Okay, so the myelinated neurons form white matter in the brain and spinal cord, while the cell bodies and unmyelinated neurons form gray matter. So white matter in the brain and spinal cord, cell bodies and unmyelinated make gray matter. Okay, so white matter. All right, so those are myelinated cells, so myelinated neurons. So that means that things got to go fast, right? And the unmyelinated, which is the gray matter, things are a little bit slower, right? So white matter fast, gray matter slow. Myelinated nerve fibers are covered by neurulema, which along with Schwann cells promotes regulation of the nerve fiber after trauma. So it gets hit, it's its healing process. All right. We have here another picture of the nose of Ranvier, the neurulema, which is right here on the side of it. Okay. And then we have the medullary sheath and the axis cylinder. Okay. So what this picture is just showing you where the neurulema is. Unmyelinated nerves lack a myelin sheath. Okay, these will not be regenerated after injuries. So myelinated nerves, they can be regenerated. Unmyelinated nerves cannot be regenerated after they are damaged. So what does regeneration mean? That means grow again, right? Be fixed. All right, multiple sclerosis. So in one of our assignments, you're asked a question about multiple sclerosis. Here's what you need to know about it. Okay, the disease multiple sclerosis causes hardened tissue to form on top of the myelin, affecting the nerve transmission. The more scar tissue there is, the worse the symptoms of MS. Okay, so taking a look here, so this is what a healthy neuron would look like. Here is what a damaged myelin or Schwann cell would look like. So there it is taken apart. All right, you have all the scar tissue on top of the neuron, And so what happens then, if these are supposed to, so if the information is coming from the cell body and it's going down, it's going fast, 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 and then it slows down, slows down. So what happens then is that the information is slowed down. So if you're supposed to move fast, then it slows down. So it causes numbness, and it causes the body not to react as fast as you're asking it to. So things that can happen are slowed speech, shaking of the hands, involuntary. You're not moving your brain. Your brain is asking your body to do something, and the information is not getting there as fast. So if it was a highway, like if you're thinking about like a road, the road would be broken and you would have to take a detour, or you would have to travel, let's say if it's a paved road and then all of a sudden it's not paved, you need to slow down because the road is a lot bumpier. Paved roads, you can go faster. So if you want to think of it like that, that's what MS does. It breaks up the pavement, and and the information slows down, or you slow down. Remember, when you're explaining what multiple sclerosis is, that you're always, or explaining things in general, that you're always, this is what happens, and this is the result. Make sure you're always giving the result of what, because what happens is when you're asked a question, it'll ask you how to apply it. So it's one thing to know what multiple sclerosis is, but what's the result is, and this is just going forward for everything. So just think of it like that. I know what it is, but what is the result of this? All right. So here's some videos for you to watch. Okay. They are in the course. They're in lesson 2. Take a look at them. Learn a little bit about multiple sclerosis. It's a really debilitating disease, because it's your body is actually attacking it from the inside, and it's treating the myelin like it is, like it's an invader. So it's called an autoimmune disease, when your body attacks itself. All right, so neurons are classified into 4 types. 4 types of neurons. We have the anaxon neurons, okay, found in the central nervous system. There's no axons, and they actually don't know what its function is, which is pretty crazy, right? So I don't know if you want to learn, research something. All right. Next, we have the unipolar neurons. They have a cell body off to one side. So unipolar, off to one side. Axons and dendrites are continuous, associated with receptors sensing environment. So the sensory neurons. These are sensory neurons. So sensory neurons are unipolar. Then the bipolar neurons, okay, so those are single dendrite and axon with a cell. Okay. The body of the cell is found in the middle of the axon. So bipolar neurons, the cell body is found in the middle of the axon. So this is how you're going to be able to identify the different types of neurons. So they're rare. The rods and cones are found in the eye. So bipolar, rare, eye, okay, found in the middle of the axon, not as common. Right? So multipolar neurons, several short dendrites and a single long axon with one or more branches. Okay, so several short dendrites and a single long axon with one or more branches. There's the most common. Okay, so activate glands or muscles. They're the motor neurons associated with neurons. So like the 2 most popular ones, right, were multipolar neurons and unipolar, right? So we have sensory and multipolar are the 2 most popular ones. All right, so this is the anaxon right here. All right, so this is what it looks like. We got the unipolar, we got the one off to the side, and then we have the bipolar. All right, so in the middle, okay, and then we have the multipolar, which is the most common. So this one, this one is the motor, and this one's the sensory. Okay, the bipolar one was the rare one where it's in the rods and cones. Okay, so here we go again. we have the unipolar here, which is a sensory neuron, and then we have the multipolar here, which is the motor neuron. So as you can see, they have the axon that comes down. They have the Schwann cells on them. These are the most common of the two, and this is the difference. What they look like, right? So here, if you see something like this, right, with the unipolar, you know that it is a sensory neuron. And if you see a motor neuron, I mean, it's built like this with the cell body on top right here, then you know it is a motor neuron. All right, conduction of nerve impulses. A nerve impulse, so AKA an action potential. So whenever you hear a nerve impulse, just know that it's action potential, has both a chemical and electrical component. So first is an electrical event. Okay, a resting nerve is said to be polarized. A resting nerve is said to be polarized. So if it's resting, it's called polarized, period. It has a relatively negative charge on the inside and a relatively positive charge on the outside. Positive on the outside, negative on the inside. Okay, you need to know that. All right, so conduction of a nerve impulse, again, aka action potential, has both a chemical and electrical component, as I mentioned. All right, and it can be measured through an oscilloscope. All right, so here we're taking a look at this right here. We got the cytoplasm in here. Okay, we have potassiums on the outside, and at rest, it is positive, it was positive, positive on the outside and negative charge on the inside. Okay, so at rest, negative charge on the inside, positive on the outside. We know at this point right here, it is at rest, right? So it is an action potential. It's known as polarized. The difference in this charge is called the resting potential or the membrane potential. This charge difference is caused by the relative concentrations of sodium ions and potassium ions. So relative concentrations, that means that whatever the concentration is, so it's relative to each other. There are three components to an impulse. Before these components begin, the nerve starts at the rest or polarization. Got a little spelling mistake there. All right, so polarization. In the resting membrane, the neuron is positive on the outside relative to the inside, or negative on the inside relative to the outside. The resting membrane is said to be polarized. Okay, so it will look like this. So the outside, okay, so you're going to see you'll see questions like this. There's been lots of diploma questions like this where it'll show you positive on the outside and negative on the inside. You need to know that that is at rest. Okay. A resting membrane potential is a difference of 70 millivolts compared to a battery of 1500 millivolts. So the resting potential difference, the difference between these are negative 70 millivolts. Okay, another important number that you should know. A polarized membrane has more sodium on the outside, so sodium on the outside of the axon than potassium on the inside. And then the sodium would be, or the potassium is on the inside. All right, here again is another representation of what a polarized membrane looks like. You will see questions like this. Polarized membrane, we know positive on the outside. So remember, polarized positive on the outside, negative on the inside. Okay. So what happens then is the resting potential, as we mentioned, okay, these will change, and then there's channels. Okay, we're going to get into these. There's specific channels that will move the potassium in one direction and the sodium in the other direction when they need to change after they change or they're getting depolarized. Okay, so there are three contributors to the charge separation of a resting cell. The biggest contributor, the sodium potassium pump. Okay, So there's an item that will move these charges back through these channels, and that is called the sodium-potassium pump. Okay. The charge difference is caused by the relative concentrations of sodium ions and potassium ions. So the charge difference will be the change. Okay. So these will change from positive on the outside, moving inside, to negative, moving to the outside. Okay. Actively transports 3 sodiums out. Okay. So 3 sodiums go out of the cell for every 2 potassiums that come into the cell. Therefore, there is a more positive charges on the outside of the cell. Okay. So it goes from positive to negative. So it's moving. All right. So as the 3 sodiums out of the cell for every 2 that goes in. All right. Large negatively charged proteins present inside the cell. They are so large that they cannot pass across the membrane. The membrane is nearly impermeable to smaller ions. Therefore, there are more negative ions inside the cell. A polarized membrane has more sodium on the outside of the axon than potassium on the inside. So there's more sodium on the outside. So when the potassium pump kicks in, sodium comes in, potassium goes out. So you get that change. So you get that depolarization, which we're going to talk about. Now, does this sound confusing? 100%. Is it tough to get your mind around? For sure. This is something that you need to work through on a regular basis. Believe me, this is taught in university. Tough concept. But if you can get your mind around it, it takes time. All right. This takes time. So don't be like, oh my God, what is he talking about? It takes time to understand the way that this process works. Okay. So again, here's another picture of it. Resting membrane potential. All right. We've kind of gone over this quite a bit at this point, but it's okay because it's going to take a while to sink in. So we have the extracellular fluid on the outside. Okay. So it's more positive. All right. So we have more positive ions out here. Right. And we have more negative on the inside. All right. And as it depolarizes, because we're at polarized, so depolarization means that the charge is moving across the axon or down the axon. Okay. This concentration will change. A nerve impulse is a series of electrical signals. Okay. So this is where the electrical / chemical combined. There are 3 components to an impulse. We have the polarized resting state, which we just talked about, okay, in length. Polarized, positive on the outside, negative on the inside, all right? Then the 3 components of an impulse. So please note, the polarized is not the impulse, right? That is just things hanging out. So it's like sleeping. I'm not doing anything, just hanging out. Polarized, positive on the outside, negative on the inside. Then the impulse happens. So then there's 3 components at that point. Depolarization. Okay, so it's going from polarized to depolarizing. Okay, so resting, polarized, depolarized, then repolarization. Okay, then a refractory period. So basically, it's resting, fires. Okay, depolarization, it fires, then repolarization. It kind of gets back to where it used to be. Okay, refractory period. Think of it refract, rest. Okay, refract is rest. Okay, so polarized, fires, repolarization, coming back to where it was, refractory, resting, nothing can happen. Okay, short form of that. So let's go into the long form. Depolarization. So the stimulation of the neuron by electrical discharge or by application of certain chemicals causes sodium gates to open. Okay, and therefore the axon membrane allows sodium to pass through. Okay. So sodium goes through the gates. All right. Sodium rushes into the axon, so down the concentration gradient, which causes there to be a positive charge on the inside. So sodium goes from the outside to the inside, making more positive on the inside. Potassium goes out, okay, making it more negative. So then it's depolarizing. Negative is going down the concentration gradient, down the axon relative to the outside. Okay, so Sodium goes in. Electrical signals going down the axon. Negative is going out. So sodium comes in, potassium goes out. This reverses the membrane potential from minus 70 millivolts to positive 40 millivolts with respect to the outside. Okay. This membrane is said to be depolarized. So as it's traveling down, it's going, so it's starting at positive. So this is showing that it's positive. So they're switching spots. Okay. So it's positive inside, go outside. So it means that it's depolarizing down this axon, down, down, down. Direction of impulse is going this way. Okay. So the next one to go down will be here. And so the positive would go, or the negative would go up, positive would go down. So that's going inside the membrane. Inside the membrane, negatives going outside. So they're just transferring the charge, and the pulse, the direction of the pulse is going down the axon. Okay, so it's going from the cell body down the axon into the next cell body, right? Being transferred. Once the sodium has rushed in, it is to its equilibrium. The membrane becomes impermeable to them again, and the gates close. So it comes in, sodium comes in, gates close, it maintains it. Everything's changing as it's going down. Boom, close, and then it maintains its firing. So as it's depolarizing. Okay, so as it goes through, it finishes, transfers all its information as it's going down the axon. Okay, once the action potential has peaked, the Na gates close and the potassium gates open. Okay, this has the effect of making the outside of the membrane more positive again, but with the sodium and the potassium on the wrong sides of the membrane. Okay, so everything's switched. The sodium potassium pumps will then kick in and exchange the sodium, the sodium and the potassium to restore the resting potential. So it depolarizes, gets to its limit. Okay, now it's got to start readjusting its levels of sodium and potassium. So potassium starts coming back in, sodium starts coming back out. So kind of just like it, it's not quick. It's like it's a lot slower than when it was depolarizing. Okay, this process of polarization, depolarizing, and repolarization takes about 2 milliseconds. Okay, which is really fast. All right. Again, so the information is going from here, firing down, being transferred over here. So this starts at the presynaptic neuron and then goes to the postsynaptic neuron, and then these become the presynaptic and then postsynaptic. So it just, the information is flowing down, right? So we have this arrow here showing you that the information is coming this way. Okay, so it goes from resting to depolarization, so polarization, so resting to depolarization. Okay, then repolarization, and then we get to the refractory period. Okay, and so if we're going to put it on a graph, this is kind of what it looks like. This is what it looks like here. Okay, so right here we have resting. Okay, so this is resting right here. Sodium channels open. Boom, depolarization. Boom, repolarization. And then we get to this point here where it's refractory period, which we haven't talked about yet, but nothing can happen at this point until it gets back to resting. Okay, so this refractory position here, this is where it rests. So resting, so polarization, boom, sodium channels open up. Here we go. Sodium channels open up. Man, flowing in, bam, it's fast. More sodium channels open up, bam, fast. So here in the depolarization, the sodium channels are opening up. Now they start to close. Potassium channels open. Boom, it repolarizes, and then we get to the point where they're closed, and then everything where the refractory period begins, and then it gets back to resting. Okay. These numbers here on the side, they're always important to know what's happening here. So it says 30 plus millivolts here, but we know that this goes to 40, right? All right, here's another representation of exactly what's happening. Okay, so polarization, just hanging out, boom, depolarization, boom, repolarization, hyperpolarization, also known as the refractory period. Okay, so you'll see both of these, and then it goes back to its resting potential, which is also known as polarization. All right, here we have it on the graph where it's telling you where the millivolts are. So sometimes you'll see this representation of 30. It can be 40. It's kind of it depends on who's writing it. Some say 40, some say 30. Okay, just be wary of that. And then time in milliseconds. So this takes one. All right, 1, 2, 3, 4 milliseconds to where it's back to its resting potential. Okay, within milliseconds, the membrane then actively pumps out the sodium to restore the resting potential. All right, here we go again. Boom. See, this one said 40, the last one said 30. Okay, just know that it's in this range. They haven't exactly nailed down exactly what happens. Okay, so know it's in that 30 to 40 millivolt plus millivolt range. Okay, so this point right here, 0.5 milliseconds from here to here, 0.4 from the start of depolarization to the end of the refractory period or the hyperpolarization. It takes 4.4 milliseconds. Okay, this is not measured because it is resting. It is not doing anything, so it's not being measured how fast this goes. Okay, so polarization is not measured in time because they're not always firing. Okay, you will see pictures like this in tests and exams and things like that. All right, they love this, showing the polarized, the plus and the negative and how it switches. Okay, I even have that in one of my quizzes. All right, so depolarization, we have the sodium coming in, boom, 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 boom. What's going out? Well, it's the potassium is going out. Okay, and as it goes down, positives go, sodium gets pumped out of the neuron, depolarize. So during depolarization, more positives are coming in. All right, so it flows, it's negative, and flows. As it flows, positives coming in, and then depolarization is coming out. Or sorry, on repolarization, the positives are going back out. So this is showing repolarization at this point, right? So this is depolarization as it's going, and this is showing repolarization. All right. Refractory period. So remember that nerves are bundles of nerves. So when one neuron fires an impulse, okay. Remember that nerves are bundles of nerves, and so when one neuron fires an impulse, it causes the neurons around it to fire as well. So it's firing, it's continuous. This wave of depolarization, okay, the action potential moves along the axon away from the stimulus. This is the nerve impulse, so it's going down the nerve, okay? Until the resting potential has properly restored, a second action potential cannot be conducted along the axon. This is refractory period. So we have polarization, we have depolarization, we have repolarization, and then that refractory period that I showed you, that where it kind of dips down and then comes back and then comes to level. Okay, that is the refractory period. Nothing can happen until it's back to resting potential or polarization. Okay, it is at least 1 millisecond long due to the fact that for this time the membrane cannot be made permeable to sodium. So these ions cannot be depolarized. So it just means it's fired. So if it made permeable, solid can't, or permeable, so that means that it's flowing, and as it's flowing, these cannot be depolarized. Okay, until it's set. So basically, fires, gets set. Once it's set again, then it can fire again. Boom, fires, gets set, can do it again. The stronger the impulse, the longer it takes for the nerve to recover. So the stronger the impulse, the longer it takes for the nerve to recover. So remember I was saying that there's kind of like, 30 millivolts plus and 40. It depends on how depends on what is being fired. And again, the refractory period, again, could be longer if it is a very strong fire, okay, or a very strong impulse. All right. So again, here's what it kind of looks like when we're looking at it. We're trying to get a visual of it. Okay. So the current flow. So we have the cell body here. We have the trigger zone. So the impulse is firing. We have the sodium that's going in, right? Current flow due to the channels opening of the sodium is opening, so it's coming in. Boom. Okay. So it shows 1 millisecond right here. Leading edge of the action potential, so it's going down here. All right, then it moves to 5 milliseconds. All right, 10 milliseconds. So as it's going, so 1 millisecond, 5 milliseconds. So that's a continuous conduction. So let's take a look at that again. So it's going, current flow to channels, going, moving. All right. So sodium moves in as it goes along. Okay. In nerves wrapped with Schwann cells, myelin sheath, the nerve impulse jumps from one node around VA to another, greatly speeding up the transmission. Okay. So what happens is it's moving from here and then it'll jump. Boom. It'll jump. So that's how it makes it faster. So it goes from here, right? From here to here to here. All right. So that's how it moves faster. It's called saltatory conduction. Saltatory conduction. Okay. So kick it again. Here, jump. All right. So it can move in a greater time, a greater distance in a shorter time frame. Once the impulse has traveled through the entire length of the axon, it reaches the axon terminal. The axon terminal is in close contact with the dendrites of another neuron. The space between the axon terminal of the presynaptic neuron and the dendrites of the postsynaptic neuron is called the synapses. So the presynaptic neuron, which I've pointed out before, is where the information is coming from. The post is where it's going to. Okay. The space in between those two is called the synapse. These vesicles store chemical neurotransmitters, which act as messengers that move through the synapse to help carry the impulse from one neuron to the other. So these vesicles, okay, so the end, store chemical transmissions, so the neurotransmissions, so they kind of releases it. Like, well, if you ever see videos of it, which there are in the course, it kind of looks like it's just flowing out, which acts as a messenger. So it's like taking its message and letting it know, hey, time to go. I'll carry the impulse from one neuron to another. So poof, pulse. It looks like this, right? So there's a synaptic cleft, kind of like that. Let's see if I can get that right in there, right? And then the chemicals are released. Boom, the information is taken in, and then it's moved on to the next neuron. Okay. So here again, a picture better than my hand. Synapse is the tiny gap between the neuron and the next cell neuron. Okay, so it's a tiny gap. So you can see the synapse right here on the bottom. So if you're looking at this bottom picture, you can see how close it is, right? And then these are the neurotransmitters. So they're representing the chemicals that are moving from the synaptic cleft, or from the synapse area. Okay, and then the information is getting transferred. Boom. So one information says another, then it goes along. So it's going this way, right? Information is going this way. So it's going from here, hop, right? So it's going jump. Information goes to the ends, and here at the ends, the information is passed from this guy, from this axon to this guy, through the synapse in the synaptic cleft. Okay, so elements of a chemical synapse. So we have the presynaptic neuron. Okay, so this is where the information is coming from. We have the synaptic vesicle, okay, where it's releasing its chemicals. Then we have the bulb, okay, synaptic bulb. Then we have the cleft, so that's the space in between. Then we have the postsynaptic neuron, where it takes in the information. Okay, so on the other side, so on the postsynaptic cleft, there is neurotransmitters, okay, that are taking in that information. So think of them as gates. It takes in that information, then it passes it on. These are the sodium. So sodium are considered the sodium, are the green ones. And these guys here, which are the calcium, this helps the transmission, okay? Then we have the plasma membrane, which is the outside of it, of the neuron, which is just doesn't allow, so it keeps it from being permeable until it is ready to take in the information through the chemically gated channel. Okay. All right. Here's another representation of what's happening. Okay, so the action potential is coming along here. We have the postsynaptic cleft. Okay, it's coming along here, and action potential reaches the voltage gated calcium channels. So the action potential is coming through here. We have the voltage gated calcium channels. This opens up the calcium channels and allows calcium ions to flow into the presynaptic cleft. So action potential, calcium channels, boom, opens it up. Then the calcium ions induce the neurotransmission vesicles to fuse to the presynaptic membrane. Okay, so information goes here. All right, then the neurotransmissions are released and diffused across the synaptic cleft, okay, to the postsynaptic cleft. So it's being released right here. So see how it's opening up. So if we move back here, information, the calcium is outside here, calcium ions, these open up, calcium ions come in. So we have the calcium ions on the outside, their action potential reaches the gates open up, the calcium comes in, the calcium tells the neurotransmitters are then released from the synaptic cleft so that everything opens up, information goes out into the cleft. Okay, so the information released, boom, and the neurotransmission bind to the neurotransmitter receptors. So these are receptors on the outside here, and then on this postsynaptic membrane, this depolarizes the membrane and induces an action potential to the postsynaptic neuron. Okay, so at this point right here, this is in resting. Okay, so it's polarized, and then it receives its information. Then it's depolarized, and the information keeps going down the gradient, right? So here is that information as it's being charged. So it's going from the start here, boom, it's coming, keep coming. All these things happen. Calcium comes in, lets it knows to open it up. Okay, it opens it up. Neurotransmission diffuse across. Boom, saying, hey, here comes stuff. You got to open up your gates, and then bang, then the electrical transmission is moved down, and polarization goes from polarized to depolarized. All right, that is it in a nutshell. All right, test yourself. Okay, and can you? All right, so don't do this right now, but at some point, draw a picture or describe how one neuron passes an action potential to another neuron. It is important that you know how that is happening, okay? And then you'll need to know what an excitatory neurotransmitter transmitter is. Or do you know what an excitatory neurotransmitter is? Do you know that? Well, if you don't, I'm going to tell you right away. Anyway, so be able to draw a picture and describe how a neuron passes an action potential to another. So it is a very long process, okay? From where it starts, like you think about it, it's already taken me 30 minutes to try to explain this to you, and I still have more slides to go through. If you can stick around, stick around. If you can't, I understand. Go. I wasn't expecting to take that much time on it, but it is what it is because it's a complicated concept. So when I was learning it a million years ago, what I had to do when I was learning about this was I actually had to break it down into very small pieces in order to see the big picture. Okay, so draw this out, learn how, try to explain it to somebody. If you can learn how to explain to somebody, that basically means that you know how to do it. Okay, draw it out, explain it, make multiple flashcards on this, learn it, go through it, test yourself, test your friends, have people test you on it. It's important to understand. It's a complicated concept, but once you get it, it will make sense. All right. So there are two types of neurotransmitters, as I was mentioning. Okay. One, this is excitatory. So the acetylcholine is the excitatory neurotransmitter. All right. It causes the sodium channels of the postsynaptic membrane to open the enzyme. So cholinesterase or acetylcholinesterase is released after ACH to break it down so the ACH doesn't continue to try to stimulate the postsynaptic neuron. Okay. So it gets released and then ACH comes in and breaks it down so it doesn't just keep being open, right? So it doesn't keep wanting to get fired. Okay. Some of the ACH is also taken back in the presynaptic neuron to help clear it out of the synapse. So insecticides block cholinesterase, so that which eventually ends up killing the insects. Okay. For alertness, learning, and memory, and is being researched in its possibility role in Alzheimer's disease. Okay. Neuroepinephrine or epinephrine is also an excitatory neurotransmitter that upon release and stimulation of the postsynaptic neuron is removed from the synapse through the reuptake. So, as in homeostasis, if there is excitatory, there has to be an inhibitory. Okay. So the inhibitory neurotransmitters, so like GABA or gamma aminobutyric acid, are known as GABA, hyperpolarization effect. So hyperpolarization, do you remember what that is? Okay, so that's the refractory period. So that makes it stay unable to fire for an extended period of time. So when we were looking at the graph, so going up, going down, and then it dips kind of down. So that dip kind of down phase before it gets to the polarization, so the resting, that'll extend that space. Okay, these neurotransmitters make the postsynaptic membrane more permeable to potassium rather than sodium. By opening more potassium gates and letting potassium out of the cell membrane, increasing the number of ions outside the cell relative to the inside. This causes a calming effect, bringing the neuron further from its threshold potential, since the outside of the neuron is even more positive than normal. This makes it even more difficult to generate an action potential in the postsynaptic neuron, so it has an inhibitory effect on its activity. So if it's supposed to be firing and goes fire, fire, what it does is it goes fire and then the pause, longer pause. So it slows everything down. Okay, problems with GABA are often found in people with epilepsy, Huntington's disease, and some people with sleep disorders. All right, speed of the nerve impulses. Different nerves can move at different speeds depending on their function, location in the body, and their axon diameter. Okay, diameter is how thick things are, right? Myelinated neurons in slate have a faster transmission speed than unmyelinated neurons. C nerve fibers are unmyelinated and have a smaller diameter, and they move the slowest. A delta and A beta nerve fibers are both myelinated, but A beta nerve fibers have larger diameter. Okay. All right. So an A beta nerve fibers carries impulse relative to touch. So an A delta nerve fibers carries impulses relative to initial pain, temperature, and C nerve fibers carries impulses related to pain, temperature, and itch. All right. And this is just showing you how fast a C nerve fiber will move compared to walking. Okay. Running an A delta nerve fiber. Okay, so it's more like an Olympic sprinter, but not as fast as a bicycle. And then an A beta nerve fiber, it's not as fast as an Asian swift bird, but almost as like an airplane is faster, but like a quarter, I'd say, is a third faster. Okay, so 2/3 as fast as an airplane. Okay, so speed miles per hour. So a beta nerve fiber can travel anywhere between 220 miles per hour. Okay, airplane travels at 350. So you see that, all right? All or non-response threshold. So a stimulus must be large enough to start an impulse, must reach a threshold level, a minimum stimulus to produce a response. Okay, so it either reaches that threshold and something happens, or and nothing happens. Each axon will have its own threshold level. Okay, so increasing the stimulus strength does not increase the impulse strength. A neuron will either fire or not fire. It's called the all or non response. The intensity of a stimulus is instead detected by an increase in the frequency of the nerve impulses. So example, a warm glass or a rod, slow rate. Hot glass, rod, frequency of impulse is greatly increased. So if I grab a warm glass, all right, I can handle that. If I can, a hot glass, it's more of a shock, right? Oh, that's hot. It's called the withdrawal reflex. All right, so the measurement of charge. So yeah, it doesn't fire, doesn't fire, doesn't fire. Boom! Oh yeah, it fires. Okay, that's just the representation of it. In the diagram above, neurons A and B are excitatory neurons, and neuron C is an inhibitory neuron. So it's try and fire, try and fire, try and fire. It's inhibiting. So we need both the A and B. Bang! Fires it. Okay, in order to stimulate D. Okay, so get this guy going. So we have A and B and C. So two excitatory and one inhibitory in order to stimulate neuron D. So get this guy right here. A and B must be fired at the same time. Boom, boom, fire at the same time. And C must not be fired at all. C has the effect of canceling out A and B since it is inhibitory. So if these two fire at the same time, boom, D will go. Okay. If A and B fire and C fires, nothing happens. If B just fires, nothing happens. If A just fires, nothing happens. Okay. A and B need to fire at the same time, and C cannot fire in order for it to move. Some neurons require impulses from many other neurons in order to fire. This is called summation. Okay, so multiples firing at the same time. All right. So, for example, these all need to fire in order to fire this one. If all three of these don't fire, then this one doesn't fire. Okay. So this is called divergence. So one, it diverges. So it fires, boom, then it fires continuously. Here, a whole bunch of all these need to fire in order to get this one to go. Okay. So if one of these are missing, one of these don't fire, it doesn't happen. All right. Sorry, I'm going a little bit fast about here. Factors influencing the speed of conduction. All right. So the diameter of axon. The larger the diameter, the faster the rate of conduction. Myelinated axons, okay, so the myelin stops, ion moves across it, therefore action potential are only generated at the nodes around VA. This is called saltatory conduction. Reflex arc, okay, so what's a reflex arc? Okay, so it's the simplest neural pathway since reflexes are autonomic response to sensory stimuli that occur without the brain stimulation. Reflexes are involuntary, okay, autonomic rather than voluntary. Okay, so reflexes are involuntary rather than voluntary. Okay, so for example, catching a baseball is a voluntary action. It is a learned action. Blinking your eye is involuntary. Okay, or tears coming down your eyes or coughing, involuntary. These involve parts of the central nervous system and peripheral nervous system. The pathway of neurons that are involved is called the reflex arc. Okay, how do excitatory and inhibitory neurons work? What is the reflex? Give an example. These are things that you should be able to answer, okay? So make sure that you're able to answer these questions. What factors affect the speed of a neurotransmission, right? Be able to answer these questions. We got 3 slides left. Okay, the knee-jerk reflex, okay? 7 essential components. Stimulus, the rubber hammer strikes a tendon just below the knee, okay? So the infrapatellar tendon. So everybody knows this reflex, right? You hit the knee in a specific spot and your knee kicks out. right? So they have the receptor, so the stretch receptor and the quadricep muscle. Okay, and then the sensory neuron on a unipolar neuron attached to the stretch receptor takes in the information into the central nervous system. So it gets hit, information comes in. Okay, then the interneuron passes the message through the message to the motor neuron from the associated sensory neuron. Okay, that's what it looks like. And then the motor neuron, then the sensory neuron synapses with the interneuron in the CNS. The motor neuron takes the information away from the central nervous system to the peripheral nervous system. All right. So then you have the effector, so a muscle or quadricep that contracts in response to the information. Response, then extension of the lower leg. Okay, so here it is. Okay, so the stimulus, the information goes in through the afferent sensory nerves, then it comes back down, and then it lets it know, hey, I got a kick. Okay, so it goes in, through, around, kick. All right, no thought goes into this, it just happens automatically. All right. Sorry I had to move a little bit fast on the end there. What should I do now? I've kept you 7 minutes later than I was supposed to. All right. So review your notes. Okay. So you may need, I may have moved a little bit too fast. You may not have been able to write down the information. You're going to have to take this video and you have to go back and fill in your notes. That's fine. You can just speed forward or you can watch it again. Maybe it'll help you. Okay. And review your notes. The information that we took in today, complicated. Okay. That's action potential. Always gives students trouble. Make sure you write down. That's why I took so much time with it. Okay, so right now what you should do is you should go back. So after I'm done, review your notes. Just don't shut it down. Review it, go over it, see if you can explain it. Draw those things out that I've asked you to do. Learn it, go over it again. Okay, maybe even do it again tomorrow. Go do your lessons if you haven't done your lessons already in this. Okay, while it's fresh in your memory, go through the notes. Okay, complete module 1 lessons. We have 8 days, as I mentioned. You got to go through the course content, textbook videos, and complete the assignments. Okay, and by all means, please enjoy the rest of your week. That's it for me. I'm done. Have a great day. Have a great week. Look forward to seeing and talking to you next week. Bye-bye. If you have any questions, stick around. I will be more than happy to answer them. Mr. Sloniski out.